What are hormones if I ask you? The answer should be, these are chemical messengers. These are chemical messengers. Can you name some hormone, madam? Can you name some hormones? Can you name any hormone? Please. My God. May God be with you. Huh? You are blank. You are black, I can understand. How can you be blank? You can't name a hormone. Try. Huh? Melano?
Melanotropin.
Melanotropin. Is there any hormone by this name? I have not heard enough. Maybe my knowledge is insufficient. Now, this is a criminal offense. If you can't say a hormone, this is a criminal offense by the Department of. Correct? So what we will discuss on, we will mostly discuss on the mechanism of action of different types of hormones. Now that we will study under these headings. Okay, what is that? Mechanism of action, hierarchy, then feedback loop, second messengers, and lastly signal transduction. Okay, signal transduction. What are the features that makes a chemical hormone? What are the features that makes a chemical hormone? What are the features? It should be secreted by the endocrine gland. It should be acting on a distant organ. Yes, that is, there is a question mark. There are few which acts in the near vicinity of the secretion. Those we call, yes, local hormones. Okay, exceptions, right? What is that? They are required in a very small amount and they can be regulated by feedback mechanism. By feedback mechanism. That means all the endocrine glands must be under some regulatory feedback loop. Just for your query, if at all we have any glands in our body or not. Do we have any glands? See? Okay. Do you have all the glands in your body? Shown here. Which one you are missing? Very good. And which one you are missing, madam? Very good. Very good. Slowly you will learn we missing nothing. We miss nothing. We have everything. It is not developed. It is not developed. Anyway. Classification of hormone. Very, very important. This is a classification which is used mostly, but in your book you will find they are classified in two classes. Group 1 hormone and group 2 hormone. That's all. 1 and 2. What is the basis of the classification? What is the basis of classification? These were previous. Basis of classification is group 1 and group 2. Group 1 is lipid-soluble. That means these hormones. Number 1. And they do have receptors inside the cell. Group 2 hormones, they have receptors on the cell surface. Mostly they are peptide in nature or they are derived amino acids. So for example, T3, T4, FT3, FT4, etc. Okay? So by chemical, if you see chemically, they are of 3 types. Either they are polypeptides, or they are steroids, or they are amino acid derivatives. All trophic hormones are peptide hormones. See, FSH, LH, ACTH. Okay? But the product of these topical hormones are steroids. Okay? See, ACTH is the cause for which these are secreted, adrenocortical hormones. You have studied the adrenal gland cross section, huh? Adrenal cortex cross section পড়েছো? তিনটে লেয়ার আছে। ড্র কন্টিনিউ। মিনালোকর্টিকয়েড, গ্লুকোকর্টিকয়েড, সেক্স স্টেরয়েডস, না? তিনটে ভাগ থাকে। অ্যামিনো অ্যাসিড ডেরিভেটিভসের মধ্যে পড়ে এপিনেফ্রিন, নরএপিনেফ্রিন আর ডোপামিন। এই তিনটেকে একসঙ্গে কি বলা হয়? ক্যাটেকোলামাইনস। ঠিক আছে? তাহলে এত হরমোন থাকতে ও কি বললো? কিছুই না। এতগুলো হরমোনের তোমরা নাম এখনই জানো, এর মধ্যে বিশেষ কিচ্ছু নেই। সিসিকে পিজেড, ফিজেড, গ্যাস্ট্রিন কিচ্ছু বলিনি, which are uncommon. Now this chart shows all the hormones which are secreted under the action of trophical hormones or trophic hormones. What are those? Hypothalamus, they secrete something called releasing hormones like TRH, thyroid releasing hormone, like CRH, cortical releasing hormone. Okay. They will act on anterior pituitary, they will secrete ACTH, ACTH will act on adrenal cortex, it will secrete cortical hormones like this for somatotropin, pancreas, insulin, glucagon, somatostatin, right? And then what is the organ? Liver. Which is not, you cannot read, na? This is liver. Thyroid glands, muscles, liver, and these reproductive organs, and mammary gland, prolactin. Okay. PRH, prolactin releasing hormone. The posterior hypothalamus, nervous, posterior pituitary secretes these two hormones. The medial part, the middle part, we do not have any activity. If we have, then it is a type of carcinoma. Normally we do not have MSH, melanocyte stimulating hormone. But anyway, vasopressin and oxytocin, we know. Yeah? They are secreted by neurohypophysis. All other trophic hormones are secreted by the yellow color, adenohypophysis. The green color, neurohypophysis. It is green or blue? Blue? Okay, blue. I am not colorblind. I was in confusion. What to call, blue or green? Whatever it is. Are you aware of this loop? Feedback loop. What do you call this feedback loop? Anyone? Yes, what do you call that? The question will come, explain the feedback mechanism of such and such hormone. So what you will write in the first? হ্যাঁ বলো।
এটা তো নেগেটিভ ফিডব্যাক মেকানিজম।
নাম কি এটা? এই ছবিটা দেখে আমি কি বলবো? hypophyseal gland axis. এটা ডিফারেন্ট হরমোন ডিফারেন্ট হতে পারে, বুঝতে পারছো? তাহলে হাইপোথ্যালামাস পিটুইটারি গ্ল্যান্ড অ্যাক্সিস, হাইপোফাইসিস গ্ল্যান্ড অ্যাক্সিস, এই ছবিটার নাম হচ্ছে এইটা। তাহলে দি সেম ডায়াগ্রাম ক্যান বি পুট ফর ACTH, TRH, thyroid TSH, LH, FSH, prolactin, right? Same diagram. We have to see what they have asked. If the ultimate released hormone is a peptide, then it must act through extracellular receptors. If the ultimate product hormone is lipophilic or lipid-soluble or steroids, then it should act through intracellular receptor. In both the cases, they must deliver the message Message means what to do, what to perform. Who will perform? Protein will perform. How he will perform? The protein has to be synthesized. How? With what? The tRNA and amino acid pool. Who will send the message, signal? mRNA. Who will synthesize mRNA from DNA? Transcription factors. Who will tell the transcription factors to wake up and do the work? Hormones in association with intracellular receptor in case of group 1 hormones, and group 2 hormones, second messengers. Because in group 2 class, they cannot enter the cell, so they must act through a second messenger. Suppose if I cannot enter the class and Ravina is there, I will call Ravina, Ravina, please put on the overhead projector and get the slide ready, I am coming. You will receive the phone call and do the work. So she will be acting as a second messenger, although I am outside the classroom. Right? So this is how they act. One such example has been given here for cortical hormones. The red dot shows direct feedback inhibition. The feedback inhibition can be of short loop or long loop. Short loop means when the hormone itself inhibits the gland by which it is produced. It is called short loop. When the hormone inhibits hypophysis, it is called long loop. Signal transduction. Read the definition carefully. It's a very favorite question. The first thing you'll get in the question is signal transduction. What is signal transduction? That's all. What is signal transduction? Ball. What is signal transduction?
The hormones which are whose receptors are present on the surface of the cell,
Is signal transduction.
the series events and components that take part in transmitting a hormonal signal to the interior of the cell.
বাংলায় বল। বাংলায় তো?
হ্যাঁ হ্যাঁ, যে প্রসেস অফ ইভেন্টস যেগুলো অকার হয় আফটার যেটা
অকার হয় মানে?
ঘটে 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 যখন ওই হরমোনসগুলো যার রিসেপ্টরসগুলো সেল সারফেসে থাকে, ওরা মেসেজ সেন্ড করে ইন্টেরিয়র অফ দ্য
এইভাবে বাংলা বলে হবে না। এইভাবে বাংলা বলে হবে না। কি হয়? তুই পারবি? বাংলায় বলতে পারবো। ঠিক আছে, হরমোনটাকে হরমোনই বল। হরমোনের বাংলা কি বলতো? এই দেখো, হরমোনের বাংলা কি বল? আচ্ছা, who can say what is the Bengali term of hormone? I doubt no one will be able to answer. Right? উদ্বোধক, ইনোকুলেটর নয়, হরমোন।
যখন কোনো উদ্বোধকের সিগন্যাল,
হরমোন বলেই হবে।
যখন কোনো একটা মানে হরমোনের সিগন্যালকে একটা কোনো কোষের ভিতরে যখন ট্রান্সমিট করা হয় বা যখন পাঠানো হবে, তো প্রেরণের
সিগন্যালটাকে সিগন্যাল ট্রানজাকশনে সিগন্যাল কথাটা ব্যবহার করা যাবে না। কি লেখা আছে? সিগন্যাল কথাটা একবার লেখা আছে লাস্টে, শুরুতে কি দেওয়া আছে?
শুরুতে দেওয়া হয়েছে যে সমস্ত
ঘটনাক্রম, the series of events, ঘটনাক্রম, series of events taking place inside the cell in response to a hormone is called signal transduction. মনে রাখবে গ্রুপ 1 এবং গ্রুপ 2 দুটোর ক্ষেত্রেই কিন্তু এটা প্রযোজ্য, মানে ইকুয়ালি ভ্যালিড। the series of events, যে একটা হরমোন যদি ভিতরে ঢোকে তাহলে কি কি ঘটবে, একটা হরমোন যদি বাইরে থাকে সেলের তাহলে কি কি ঘটবে, এই সিরিজ অফ ইভেন্টস গুলোকে যদি আমি ধরি তাহলে বলবো সিগন্যাল ট্রানজাকশন। ট্রানজাকশন মানে টু ট্রান্সফার the message in forward direction. Someone else will carry this. How? It will first, see, it will first has to link with what? Either membrane or cytosolic receptor. Cytosolic receptor মানে intracellular receptor, cell ভিতরে. So if it is group 1 hormone, it will bind to cytosolic receptor. If it is group 2 hormone, what will be the fate of T3, T4? Huh? T4 intracellular, thyroxine intracellular. কেন? সাইজে খুব ছোট, they are not water soluble, lipid soluble, but they are very small in size, single amino acid. That's why they can enter. Hello? So the binding of hormone, or we call it ligand or ligand, to the receptor triggers a series of events. Triggers means it initiates a series of events. একটা ঘটনা ক্রমের সূচনা করে। Signal initiator, it should be always inside the cell. Signal mediator, someone has to carry the signal from cell surface to interior. Number one. If it is cytosolic receptor, then someone has to carry from that hormone receptor complex, or HRC, hormone receptor complex, to the tissue where this is targeted, that is DNA, molecule. Then it must reach a target molecule. Mostly the target molecules are DNA. Why? Because that will bring about conformational changes in the DNA, which will open or close some of the transcription start site, which will initiate the production of some of the mRNA or stop the production of some of the mRNA, whatever is required for the function to be done. And then the protein will be either synthesized or stop synthesis. And then in presence of protein, some functions will take place, or in absence of protein, some function will shut down, and the biological function will be done this way. Clear? যে কোনো হরমোনের ক্ষেত্রে এটা খুব সহজ, একটা জেনারেল পাথওয়ে, এইটা সুন্দর করে মনে রাখতে পারলে ইউ ক্যান আনসার এনিথিং। All hormones interact with target cells by first binding to specific receptors located either on plasma membrane or as a cytosolic protein. The receptor for hormones must be linked to a component that is able to respond to the binding of hormone with its receptor. Someone will respond after hormone is bound with the receptor. Who is that? We'll see. Because signal transducers, these molecules are called in one term signal transducers. So substances that fool the responder into thinking a hormone. Okay? The substances which are like the hormone but don't have the hormonal action, they are called and agonist and antagonist. Agonist means you will fool, you will no function. Antagonist means opposite function. You stop the function. One of the signal initiator we will start with G protein. Common short note, G protein. of G protein were awarded with Nobel Prize? Very good. And they wrote a book. And they wrote a book on pharmacology. আর চলছে এটা ক্যামেরা? আচ্ছা এটা বন্ধ হয়ে গেছে. So they wrote a book on pharmacology. The name of the authors are Goodman and Gilman. This Alfred Gilman received Nobel Prize. His father also received Nobel Prize for something else. Father, like son. Anyway, so G proteins are usually found in association with cell membrane. They have three components or three different parts: alpha, beta, and gamma. Okay. Among them, beta gamma stays together and alpha stays separately. Gamma is not directly attached to the cell surface, cell membrane. Only the alpha and beta are attached to cell membrane, and gamma is attached to beta subunit. Now, depending on what type of function this G protein is going to initiate, there are different types of alpha. Beta and gamma remain same. Alpha types are different. So G protein which will stimulate some activity are provided with G alpha S, means G alpha stimulatory. G protein which perform inhibitory action are provided with G alpha I, G alpha inhibitory, right? G protein which are related with the light sensing activity in the eyes are termed as G alpha T transducers, right? So we have different types of alpha subunit for different functions done by G protein. Beta and gamma remain same. Now the beta usually, the alpha and beta gamma, you must say like this: alpha and beta gamma, because beta and gamma say together. The alpha and beta gamma, these two remain associated, closely associated when it is in inactive form. The moment some hormone binds to the cell surface receptor, the cell surface receptor usually has three domains. It's a protein present on cell surface, has three domains. Number one, extracellular domain. Number two, intracellular domain. Number two, transmembrane domain. And number three, intracellular domain. The red dot is a hormone. The green bar is a receptor, and to the receptor, this green bar, it is for the case of G protein, the receptor, suppose this is a cell membrane. The receptor has three domains. The receptor has three domains. This is and this is, Now, there is a very easy way to explain how does it work. I want one volunteer who will not feel bad upon whatever I do on my activity. So who will be a volunteer? I want boys. If you do not volunteer, I will volunteer to find someone, whoever is nearest. Whoever is nearest. Who is nearest? Now, stand here. You see, don't beat me, huh? You see how these receptors act. See, I did something with his ears, but some expression came in his mouth. I didn't do anything with the mouth. If I continue, then some other parts of the face also will show the effect. What is that? Huh? So, it is the ear who takes a beating and eyes are shedding tears. So, we perform a conformational change in the extracellular domain, which is transmitted through the transmembrane domain, which is invisible to us. Invisible. What is the link between ear and eye? Invisible. But effect is seen by the intracellular domain, again by conformational change. Now, what the structure I have drawn, it has got a specific name. This particular receptor has got a special name. It's called 7-TM receptor or 7-transmembrane receptor. So G protein is associated with 7-TM receptor. Correct? The story looks very simple, but it is not at all that simple. Now we come to G protein. Now see, initially it was all together, alpha and beta and gamma were closely associated. So when they are associated in this region, right? Now this region of G alpha has got a sitting position for GTP. The moment this beta gamma goes little far or dissociates from alpha subunit, this position is open, and then this GTP is hydrolyzed. Some energy will be formed or transferred, and this G alpha will then the G alpha will then approach this membrane bound enzyme called the purple color, what? Adenylate cyclase, otherwise known as adenylyl cyclase. Adenylyl, both are same. Adenylate cyclase or adenylyl cyclase. Now, this AC, adenylyl cyclase, is activated by G alpha. Good. When G alpha is stimulatory, S. If it is I, then it will inhibit. Correct? If it is T, it will not act on AC. It has something else, some other molecules to act on, not AC. So, same hormone can trigger different G proteins depending on different tissue. You need not to produce, suppose thousands of effects are taking place in our body. That means we need thousands of hormones. No, one hormone can perform several works by just altering G alpha protein. This is one stage where signal initiator is there. The second stage again it may bifurcate. Third stage again it might bifurcate, depending on tissue. Right? Because same thing act on all cells, melanocyte stimulating hormone. Right? Only iris becomes black, not the lens. Only iris becomes black, not the lens. So same hormone, different action, different tissue, due to different signal initiator or signal transducers. Try to remember these terms. It will be easy. So this is the process by which a G protein is activated. And the same thing has been shown, the series of events for activation of G protein has been shown in this diagram. A beautiful diagram, you can draw it. It was given by Gilman himself. No? So most authentic from the horse's mouth. Bolo. Good question. So just put one arrow between these lines. Then see, it becomes a flowchart. Put one arrow between the lines, then it will become a flowchart. Now you have diagram side by side and occurring events taking place in the other side. Any question so far? Any doubt? Just know what to read, know how to read from the class. You cannot read it here. You have to read it afterwards. But you must take home from the class how to read and what to read. And more importantly, what not to read. Remember, any doctor knows when to operate, but a good surgeon knows when not to operate. Any doctor knows what to be given, but a good physician knows what not to be given. Likewise, any teacher knows what to be taught. Very few knows what not to be taught. You must enjoy while studying. You must remember the things the way they should be studied. Every teacher has a different approach to a chapter. But these are the basics on which all understanding will, whatever you understand about the endocrinology will rely on. Wherever you go, whatever medicine you see, they have to function in either of these few molecules. They should be targeted. Yeah. এটা ব্যাটারি মনে কমে এসছে মাইকে। Now second messengers. হ্যাঁ ওটা ছেড়ে কারণ লাগবে না। Hello. Now, second messengers. Remember, they are not secondary messenger. They are second messengers. Secondary messenger নয়, কথাটা হচ্ছে second messenger. ঠিক আছে? আচ্ছা। So, molecules whose presence is a signal, মানে ওরা তৈরি হওয়া মানেই কোনো সিগন্যাল এসছে, তাদেরকে আমরা সেকেন্ড মেসেঞ্জার বলবো, তাই তো? কোনোভাবে যদি থাকে সিনথেসাইজড অর রিলিজ ফ্রম স্টোরেজ, সবসময় তৈরি হতে হবে কোনো মানে নেই, সেটা রিলিজ হচ্ছে। এখানে যেমন অ্যাডেনাইন সাইক্লিস কি করে, এটিপি থেকে সাইক্লিক এমপি তৈরি করে, ইট ফর্মস সিনথেসাইজেস সাইক্লিক এমপি ফ্রম এটিপি, কারেক্ট? Now there is an enzyme which also destroys cyclic AMP. Now there is a balance. When production of cyclic AMP is more than its destruction, that means in the cell will sense there is an increase in cyclic AMP concentration. Relative increase, not absolute increase, it is relative increase. So that will trigger in which direction cell must function. Similarly, all second messengers need not to be synthesized. They might be stored within a pool inside the cell, which from where it will be released in response to the signal transduction. Act as intracellular ligands. Cell surface receptors are called hormones are called extracellular ligands. Second messengers are not membrane bound, so they can act like messenger. Messenger means which is free to move. So second messengers are like intracellular messengers, commonly used in G protein coupled receptor GPCR, made or released by effector proteins ligand for first messenger. The general characteristics of second messenger are they're very low in amount, regulated synthesis, regulated destruction, act through other proteins. They do not perform any work. They trigger the activity of some other proteins which are responsible for the biological function. Name, write down. Each and every one you have to remember. No mercy. Cyclic AMP, cyclic GMP, calcium means calcium ion, Ca2+, and lipid derivatives. IP3 means IP3 or PIP2, inositol phosphate, triphosphate, or phosphoinositol pyrophosphate. Inositol triphosphate or phosphoinositol pyrophosphate. PIP2 or IP3, same. DAG, diacylglycerol. Many others. We'll see. Yeah? Cyclic GMP, see? Now, if you remember or if you recall, I said there are different types of G alpha protein subunits. I mentioned 3 types, S, I and T. So where this T act on? That is guanylate cyclase. In eyes, the corresponding protein for adenylate cyclase is guanylate cyclase, GC. So it forms cyclic GMP. What happens usually here cyclic AMP should be synthesized more in amount, but cyclic GMP concentration should be reduced to trigger Vision effect. That means G alpha will reduce the production of cyclic GMP, G alpha T. It will reduce the production of cyclic GMP, unlike cyclic AMP. See the cyclic AMP signaling pathway, receptor, G alpha stimulatory, adenine cyclase. E means adenine cyclase. ATP becomes cyclic AMP. Cyclic AMP stimulates PK, protein kinase A. লিখে ফেল, এটাই আসবে পরীক্ষায়। পরীক্ষায় এটাই আসবে। তো কি মনে হচ্ছে দেখতে ভালো বলে আমি ছাপিয়ে নিয়ে এসেছি? হ্যাঁ, মহা মুশকিল। কিন্ডারগার্টেনে ভর্তি হয়, যা। Substrate to phosphorylated substrate. PD মানে diasterase, which breaks down phosphate. Cyclic AMP phosphodiesterase. Phosphodiesterase, which breaks down cyclic AMP. Okay. এটা ছবি তুলে নে। এই সিঙ্গেল ডায়াগ্রামে সবকটা সেকেন্ড মেসেঞ্জার দেওয়া আছে যেগুলি জি প্রোটিন দিয়ে আসে এবং ডিফারেন্ট সেকেন্ড মেসেঞ্জার। এখানে 1 নম্বর, নাম্বার 1, the ligand is, come on, tell, the ligand is, you tell, glucagon, read. Signal initiator is, second messenger is, primary effector, protein kinase A, secondary effector, protein substances. These are the words, these are the terminologies. Next, number 2, this is nitric oxide. Nitric oxide, second is nitric oxide. Primary effector, signal initiator is G protein. Primary effector, second messenger, cyclic GMP. Primary effector, protein kinase G. Secondary effector, protein substances. What is the effect of nitric oxide? What is the effect of nitric oxide? Hey, কি বল? বস। কি বলছে? কঠিন। What is the effect of nitric oxide? কে বলতে পারবে? Anyone? এই বলো, সামনে যে মোবাইল দেখছে, মোবাইল ভেঙে দেবো আমি। Anyone? হ্যাঁ, কি বলছি? ভ্যাসো বলি, ভ্যাসো মাথা মোটা। ভ্যাসো dilator, very good. Bose, use which is given by Almighty in the upper 6 inches of your body. ওরে, এটা আছে তোর বাড়িতে? আরে, এটা আছে বাড়িতে তোর? বাড়িতে আছে, নিয়ে আসিস পরের বার, হ্যাঁ? বস। থার্ড, হরমোন এখানে উল্লেখ করা নেই, দরকার নেই। ক্যালসিয়াম। ক্যালসিয়াম কোত্থেকে আসে? সেলের ভিতরে যে ক্যালসিয়াম পুল থাকে সেখান থেকে আসে। So it acts on calcium calmodulin complex. The binding of calcium to calcium calmodulin complex. Calcium calmodulin মানে? What is calmodulin? It's a super secondary structure. It is not a specific protein. It is a part of the protein having a particular structure which will feed only with calcium ion, found in many proteins. There is no single protein which contains calcium calmodulin. There are many proteins which interact with calcium ions. They must have calmodulin complex. We'll stop here.